Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biotechnology conducted in the year 2017, and I will be discussing only the biology part. So coming to the first question. Which of the following statement is correct? Cellulose is a homopolymer of glucose. Insulin is a heteropolymer of glycine and tryptophan. Insulin consists of 51 amino acids. It is not a heteropolymer of glycine and tryptophan. Then lactose consists of glucose and fructose. Lactose consists of glucose, galactose and glucose. Then maltose consists of galactose and glucose. Maltose consists of 2 glucose units. So cellulose is a homopolymer of glucose. That is the answer. That is the statement that is correct. Then next one. Which of the following breaks an ester bond? So lipases catalyze the hydrolysis reaction of lipid ester bonds resulting in alcohols and fatty acid. So the answer here is option A. Then next one. In the isolation of DNA, removal of protein and RNA is carried out by the enzyme dash and dash respectively. So it is carried out by protease and ribonuclease. Answer is option C. Then glyceraldehyde 3-phosphate is an isomer of. So it is an isomer of dihydroxyacetone phosphate. Answer is option B. Codon bias and CpG islands are useful in eukaryotic genome too. So cysteine and guanine doublets, they are found concentrated near the gene-rich regions of the genome. Sorry. Codon bias is the phenomenon often observed in coding region of DNA for certain codons to be used over other coding for the same amino acid. So the answer here is identify open reading frame. Answer is option C. Then the dye used to track the migration of proteins in the polyacrylamide gel is bromophenol blue. Answer is option C. Then which technique can be used to detect the protein-protein interaction? So co-immunoprecipitation, yeast-2 hybrid system, FRET microarray, all of the above can be used to detect protein-protein interaction. Then transversion is a type of. So transversion is the substitution of a purine by a pyrimidine or a pyrimidine by a purine. So it is a type of point mutation. Answer is option B. Which of the following methods could be most readily employed to identify tryptophan? So ultraviolet spectroscopy can be used because the amino acid tyrosine and tryptophan, they are having very specific absorption at 280 nanometers, allowing direct 280 measurement of the protein concentration. Then next one. Which vitamin is produced by the microorganism present in normal microbiota? So it is vitamin K that is produced. So it is vitamin K2, a component of vitamin K1 synthesized by bacteria present in the intestine. Then next one. In cation exchange chromatography, the solid support should be. So it should be negatively charged in cation exchange chromatography because cation exchange chromatography more specifically uses negatively charged ion exchange resin with affinity for molecules having a net positive charge. Then next one. The term hyperglycemia refers to. It refers to increased blood sugar. Answer is option A. Then chemically, agar belongs to one of the following macromolecules. So it is carbohydrate because it is a polysaccharide obtained from the cell walls of some species of red algae, that is Gracilaria and Jellidiaceae. Then next one. Which of the following set of amino acid consists of only apolar or hydrophobic side chains? So alanine, leucine, isoleucine, phenylalanine. Answer is option A. The chromatography in which ligand is immobilized to the column matrix is. It is affinity chromatography. Answer is option B. The compound which cannot be metabolized to pyruvate, that is phenylalanine, cannot be metabolized. Then string is a database of, it is protein-protein interaction networks. Answer is option C. Then immunoglobulin that result in histamine release. This is a repeated question. It is IgE. Answer is option C. Then the slope of Km by Vmax indicate. It indicate uncompetitive inhibition. Because in uncompetitive inhibition, the slope doesn't change. Answer is option B. Then next one. Gene bank is a nucleic acid sequence database which is maintained by NCBI, that is National Center for Biotechnology Information. Answer is B. Characteristic feature of allosteric protein is cooperativity. Answer is option B. Dash has very restricted conformational freedom. So proline, it is having very much restricted conformational freedom. It influences protein architecture because its ring structure makes it more conformationally restricted than other amino acid. Then Hoogsteen hydrogen bonding is present in, it is present in bDNA. Answer is option B. In heterogeneous offspring is crossed with homozygous recessive parent, such activity is called, it is called a test cross. Answer is option C. Then aneuploidy refers to, so 2n plus 1, it represents trisomy which is an aneuploidy. So answer is option B. Then following is associated with breast cancer, BRCA1, BRCA2 or both. So both. Answer is option C. Then SOS response involves, it involves thymidine dimer formation, DNA strand breaks, lex A cleavage, all of the above. Answer is option D, that is all of the above. Hydrogen bond is formed between, so a partially positively charged hydrogen atom with a partially negatively charged electronegative atom. Answer is option B. The severely affected people who possesses most common form having more than 1000 repeats of CGT triplet, that is myotonic dystrophy. Answer is option A. Protein surface is predominantly comprises of dash amino acid residues. So it consists of polar, that is A is answer. Next one. In an ideal alpha helix, every carbonyl oxygen of an amino acid residue I is hydrogen bonded to an amide hydrogen of amino acid residue, that is I plus 4. So answer is option D. These are all direct questions. Then which of the following statement concerning protein is correct? Alpha helices are stabilized by hydrogen bonding between carbonyl oxygen and amide hydrogen of amino acid. That is correct. So proteins are the only molecules that can catalyze reactions in a cell. That is not true because there are RNA molecules called ribozymes which are also involved. Then the peptide bond is formed through a hydrolysis reaction. Peptide bond is formed through a condensation reaction and a peptide bond can be broken by hydrolysis reaction. Disulfide bonds are formed from ionic interaction between charged amino acids. So disulfide bonds are covalent bonds derived from two thiol groups. So B, C, D are not correct. Which statement about mitochondrial genomes is false? So they use genetic code different from the standard eukaryotic genetic code. That is correct. Their genes do not have introns. Okay. They probably arose as a result of symbiotic event with formerly free living organism. That is also correct. They contain gene necessary for all mitochondrial function. That is wrong. Because mitochondria are not self-supporting entities but rely heavily for their functions on imported nuclear gene products. Okay. So even though they have got their own machineries for producing proteins, some proteins are synthesized in mitochondria, but majority of them are synthesized in the nucleus and then transported to mitochondria. Then, which of the following has the highest percentage of modified bases? So it is tRNA that is having highest percentage of modified bases. Then which of the following enzymes is the anabolic pathway given below is most likely to catalyze the rate limiting step in the biosynthesis of A6? So it is E3. Answer is option C. Which one is common compound shared by the TCA cycle and urea cycle? So it is fumarate. Answer is option C. Then which of the following statements best describes an allosteric binding site? So it is a binding site containing amino acids with aliphatic side chains. So it is a binding site that can accept a wide variety of differently shaped molecules. It is a binding site which is separate from the active site and affects the activity of the enzyme when it is occupied by a ligand. That is correct. It is a description of an active site which undergone an induced fit. That is also not correct. Then next one. BCG vaccine is used for tuberculosis. Answer is option A, that is TB. Then chemical mediated introduction of DNA into a bacterial cell is termed as. It is termed as transformation. Answer is option A. The antibiotic that does not bind to the ribosome is. So it is penicillin that does not bind to the ribosome. So penicillin kills bacteria through binding of the beta-lactam ring to transpeptidase, inhibiting its cross-linking activity and preventing new cell wall formation. So it does not bind to ribosome. So answer is option D, that is penicillin. Then monoclonal antibodies are against a single epitope. Answer is option B. The cell wall of the following is not rigid in. So archaea, it is semi-rigid cell wall. Protozoa, it is rigid cell wall. Bacteria and fungus also rigid cell wall. So the answer is option A, that is archaea. Then the amino acid that show characteristic absorbance at 280 nanometers, that is tryptophan. Answer is option D. The codon UAA codes for termination. So answer is option D. Then toxemia means it is circulation of toxins in the blood. Answer is option B. Then collagen has got a triple helical structure. Answer is option C. Rapid but non-antigen specific immune responses are produced by the innate immune system. This is a repeated question. Answer is option B. Deficiency of thymus results in increased infection in humans. Such a condition is called DiGeorge syndrome. Answer is option C. Next one. In Krebs cycle, 2 moles of glucose produce. So 2 ATPs are produced in the TCA cycle per glucose molecule. Now 4 ATPs are produced in TCA cycle from 2 moles of glucose. So 4 moles of ATP is the answer. A bioreactor is a fermenter. Answer is option B. Prokaryotic translation elongation factor requires dash for its activity. It requires GTP. Answer is option A. Then viroids consist of. So viroids are infectious agent that consist only of naked RNA without any protective layer such as a protein coat. So the answer here is only nucleic acid. Answer is option D. During the bacterial growth, the primary metabolites will be produced during. It will be produced during the log phase. Answer is option B. So thank you for watching my channel. Please like, share and subscribe. Thank you.